Hello guys, welcome back to my channel. So as you know, we are studying about the replication of DNA. Right, so in the last video, if you saw, we studied about the RNA that is used for the DNA replication. Right, so now we'll study about the entire process of DNA replication. We studied introduction, then we studied RNA. But now we'll see the entire process, how the entire process to the replication, that is the replication machines to the DNA replication. So DNA replication requires cooperation of a large number of proteins, right? So the replication machine considers of large number of proteins, and DNA replication requires large number of protein that act in concert to open up the double helix and synthesize new DNA, right? So the first problem that, so for example, this is our parent DNA. So the first problem that the replication machine faces is to open up the DNA helix and to access the nucleotide, right? So the first issue is opening up the double helix and accessing the nucleotide, right? So for DNA replication to occur, right? So the double helix must be unzipped, so it should be unzipped ahead of the replication fork, right? So what which type of replication proteins helps in opening this DNA double helix? So two types of replication proteins are there that helps in this process, right? So we'll go one by one. So first the two types of, all right, so I will write here two types of replication proteins, right? First, there are two types of replication proteins that operate together to open up this. No, so I'll write it down, that operate together to open up the double helix. All right, so which are they? So first one is the DNA helicases. And the second one is a single stranded DNA binding protein. Single stranded DNA binding protein. So these two so these two carry out the first task. How they do it? All right. So we'll draw a simple diagram. Right. For example, this is our DNA strand. So what the DNA helix do? So all right. Let me use some other color. All right. So for example, this is our DNA strand. So I will write it down here in front. DNA. Right. So the DNA helicases, this will sit on the, here for example, this is our DNA helicase. So I'll name here DNA helicase. So what does the DNA helicase do? So it sits at the front of the replication machine. So first the DNA helicases come in the replication machine. So this sits on here the front of the replication machine, and where here it uses the energy of ATP hydrolysis to propel itself forward. So DNA helicases act on the this DNA strand, for example, and what it does. So right. So for example, this is our replication fork. So this is open, and so that is our DNA helicase. Right, so what the DNA helicase do? It opens and it sits here, right? As this is closed, for example, right? And this is our DNA strands, right? So we'll name it here DNA strand. We have the two strand. After the replication opens the replication fork, right? So the DNA helicases uses the ATP hydrolysis to move itself forward, so it goes round. So it's like a ball, for example, and as it moves, it keep opening the DNA strand, or keep separating rather the DNA strand from each other. And this is how it, so for example, if our helix was here, right, DNA helicases, so it moved further, and it opened this entire part, right? So this is the first use of our DNA helicases. This is our first protein. Now the second one, single stranded DNA binding protein. So what this protein does, single stranded DNA binding protein. So I think we'll need some other color maybe. Let's see what we have. Maybe dark green. Alright, so what is this? Alright, so what this single-stranded DNA binding protein does, this protein will hang on the open DNA. So for example, DNA helicases opens the DNA, and the DNA binding protein, for example, this sits here. So we'll say single stranded DNA binding protein, right? So why it says, the name suggests single stranded DNA binding protein, means it's cling to just the single strand of the DNA. Why it clings to the single strand of the DNA? So as the DNA helicase is moving further, and it open up the double helix, right? So if the single strand DNA binding protein is not present, what will happen? This separate DNA strand will make again the base pair together, and it will be again in the structure of helix. So this single stranded DNA binding protein, what it does, it stop the DNA strand to base pair with each other. So that's the function of our single stranded DNA protein. All right, so we have our two replication machine proteins, right? DNA helicase that move further on the replication fork to open the DNA, and we have the single stranded DNA binding protein, so that stops the base pair of the strands together for joining and again forming the DNA helix, right? So now this structure itself has a problem. So for example, if you have a rope, right, as a double helix, right, DNA double helix, and if you start opening from both the sides, so this is, I'm just showing the one side of the replication fork. For example, this replication fork goes this way, and there will be the same thing happening here on the other side as well. So on this side and this side, the same thing is happening on both the sides. So from both the sides, it is opening, right? It is opening. So when it opens on both the sides, for example, when you try to open a rope with the same like a DNA double helix, so when you try open on both the sides, what will happen? On the ends, on the further ends of the rope or the DNA helix as well, so it will try to torse. There will be a literally very tension and stress over the helix or the rope you are trying to open, and it will become tangled. So for example, imagine this is the same thing is happening over this side as well. So what will happen here? So this should be going straight, but instead what it does, it can happen. Oops, sorry. right? So something like this. It will tangle or like torsional stress or tangled. So the double helix will become tangled. So to avoid this situation, there is one other protein in the replication machine. Alright, so to relieve this stress or the tension that is happening in this DNA double helix, so we have another protein, so that helps to relieve the stress and tension in the DNA helix. So the name is DNA topoisomerase. Right? This is merase. Sorry. So this protein helps in relieving the stress and the tension that happens in the DNA helix as the DNA helix opens up. So let's see. Let's use something else. Maybe if this works. So for example, like a C. Alright. So how it does. So for example, this is our, I will name this here DNA isomerases. So what this DNA topoisomerases does, so it produces a nick in the few nicks in this DNA helix. So for example, this is our helix, right? This is our helix. So what it does, this, okay, so let me make a bigger picture so you will be able to understand. So what this DNA topoisomerase does, so it produces a nick. For example, here, that's a nick. Nicks, it produces a nick, a gap. So here you can see, we'll make like this with the green dots. I will name it here nicks. Here, this DNA topoisomerase produces few nicks in this DNA helix that is along with it, right? So because of this nicks, it relieves the stress and the tension that is happening because of the opening of the DNA helix, right? And as soon as the DNA replication fork is moving further, right? As soon as it comes here, so this DNA topoisomerase seal the nick and it falls off. So this before falling off, DNA topoisomerase will seal these nicks, right? So we had our three proteins: DNA helicases, single-stranded DNA binding protein, and DNA topoisomerases. So this is three protein of the replication machine, right? So with this, now we have our DNA polymerase, right? The main important protein that helps to replicate the DNA. So, which one will use? Another color, maybe red. Let's use the red. For example, I'm making here. Almost the same. So I'm going to use the blue one. This might help. For example, this is our DNA polymerase, right? So DNA polymerase, so as this is just a 2D picture, so I'm just showing you DNA polymerase on one strand, but it actually passes on the both strands. DNA polymerase is just one, and it synthesizes or it replicates both the DNA strands, leading and lagging, just the one DNA polymerase. There are not two DNA polymerase for both the strands, right? So we have our DNA polymerase. So on their own, this most DNA polymerase molecules will synthesize only a short string of nucleotides before falling off the DNA template, right? So the tendency to dissociate quickly from a DNA molecules allows this DNA polymerase molecules that has just finished synthesizing and one Okazaki fragment on the lagging strand, for example, this is our lagging strand, to be recycled quickly so as to begin the new synthesis of the next Okazaki fragment on the same strand. But this rapid dissociation, however, would make it difficult for the polymerase to synthesize this long DNA strand, right? So if it keeps falling, so it will be hard for the DNA polymerase to synthesize this entire DNA strand produced at the replication fork. So in order to hold this DNA polymerase to this template strand for this long DNA, what we have one more protein that is an accessory protein. It is called as a sliding clamp, and it's called as a pCNA or a necessary protein in eukaryotes. So I'll show you. All right, so the DNA polymerase is held by a sliding clamp. So I will put it as a number 4 here, and we have a sliding clamp type of a protein that helps DNA polymerase to stick on the template. So I will draw a separate picture for here, for example. Alright, so I will show you the clamp, the sliding clamp. So for example, I think I'll take the pink one here. For example, this is our sliding clamp. So how it helps the DNA to move and to hold. Let's use another one, maybe blue one, whatever. Right, so the sliding clamp has two subunits, so like black and pink. It has a two subunits, right? So, and this is loaded onto DNA to serve as a tether for moving the DNA polymerase molecule. So this is loaded onto the DNA. It is loaded actually onto the DNA, right? As a tether for moving the DNA polymerase along the molecule. So how the DNA fits in this sliding clamp is actually there is one more protein that is, or if it is a clamp loader. We need a clamp loader to load the DNA into the sliding clamp. So for example, all right, so I have this one here. Okay, so so this is our sliding clamp. This is our clamp loader. All right, so the sliding clamp and the clamp loader. So the structure of clamp loader, so the clamp loader look like something like this. So the structure of clamp loader resemble a screw nut, like a screw nut with its thread. So its thread matching the grooves of the double strand DNA. So this is actually the structure of this clamp loader is actually like matches the grooves of the double stranded DNA. So it fits the DNA strand. So how the loader? So the loader binds to first to the free clamp. Right. So the question is how this sliding clamp prevents the polymerase from dissociating without at the same time impeding the polymerase rapid movement along the DNA molecule. So this sliding clamp stops the DNA polymerase to fall off from the strand, and the DNA polymerase is rapidly moving to the strand, replicating all the DNA, right? So it does, it holds it and it moves as well. So how this is done? So the three-dimensional structure of the clamp protein determined by the X-ray diffractions revealed actually, so it to be a large ring. So this is a large ring around the DNA double helix. So imagine that this is a large ring that is around the DNA double helix. One phase of this ring, so one phase of the ring binds to the back of the DNA polymerase. So one phase of the ring is binding to the DNA polymerase on the back of the DNA polymerase, and the whole ring, this entire ring slides freely along the DNA as the polymerase moves. So I'm not sure I can make that picture, but let's make this. For example, this is our polymerase, right? So this sliding clamp, so it holds the back of the, right, it holds it, this ring binds to the back of the DNA polymerase, and the whole ring, this entire ring, slides freely along the DNA. So something like this, right? So this is our DNA polymerase, for example. This is our sliding clamp. So this is holding the back of the, sorry, this is holding the back of the DNA polymerase, and this entire ring is actually sliding around the DNA. So this hold, and then the DNA polymerase moves rapidly as the DNA replication process is going on, right? So this is something how it's done, right? So the assembly of the clamp around the DNA as requires ATP hydrolysis. So how this joins to this? So this requires energy from the ATP hydrolysis, but this does not itself attaches to there. It requires a special protein complex with the ATP hydrolysis that is known as a clamp loader that I mentioned here. That is the clamp loader, right? So this clamp loader, what it does, which hydrolyzes ATP as it loads the clamp onto a primer template junction. So for example, let's see now. So we have this, and this. Oops, not black one here. We need to use blue for the clamp loader. Alright, blue, and it is with ATP. This is not ATP, but it uses ATP hydrolysis to load the clamp. How the process is done. So the ATP binding to the, so this ATP is binding to the clamp loader. ATP binding to clamp loader, right? And that opens the sliding clamp. As you can see, this is closed, and this, that opens sliding clamp. So that opens the sliding clamp, right? So the next thing is, so as this is open, should I use maybe a gold one? So this is our DNA strand, right? So now this helps the sliding clamp to join the DNA, right? So now we have our, okay, is this, This is still not closed. It is open. And the blue one. So this is, you can see this now. Now the clamp loader. So now the DNA is engaged in the clamp. Okay, we'll make it here like. Clamp. So DNA engages in the clamp, right? So this is the green one is our DNA. We'll name it here DNA strand, right? That is engaged in the clamp. And then the ATP hydrolysis locks the sliding clamp around the DNA and releases the clamp loader, right? So next step would be, all right, so we, let me make first the DNA. Weird DNA, but whatever. So we have this. Now it's together. So this is done by ATP hydrolysis. So I will just put it here. Oops, sorry. Let me put it in blue. ATP, right? And this is done by ATP hydrolysis. So ADP and the phosphate is released, right? And here the clamp loader is released. So this is our clamp loader that has been released, and I will write it down. So what happens? So the ATP hydrolysis will lock the sliding clamp, around the DNA and releases the clamp loader. The DNA and releases the clamp loader, right? And the last step is the DNA polymerase will bind to the sliding clamp, right? So lastly, then this is released. So the clamp loader is released, and here DNA I will write it down. This polymerase will enter and bind to it. So let's make the DNA first here. Oops. Let's make the DNA here. Great. So now what we'll have, I'll make it with what should I use? Alright, I will use the blue one to make the DNA polymerase, so that will bind this clamp, for example. Something like this is our DNA polymerase, and that will, so I'll name it here, polymerase. So that will bind to the sliding polymerase clamp, right? So the DNA polymerase will bind the sliding clamp. So this is the clamp loader that we need for the DNA polymerase to move along without falling off the DNA template, right? So on the leading strand, so we have right the leading and the lagging strand. So on the leading strand, the moving DNA polymerase is tightly bound to the clamp on the leading strand, and the two remain associated, and both the sliding clamp and the DNA polymerase will remain associated for a very long time on the leading strand, right? But the DNA polymerase on the lagging strand, so this is our leading strand, as the replication fork moves this way, leading and the lagging strand. So on the lagging strand, the DNA polymerase also make use of the clamp, but each time the polymerase reaches the 5 end of the Okazaki fragment, right, as we knew, right? So this is 3 and 5 and 5 and 3. So as it reaches the 5 end of the Okazaki fragment, what it does, it ends as soon as it reaches the 5 end, the polymerase releases itself on the lagging side of the strand from the clamp and dissociated from the template, right? So there are not two DNA polymerase, just one. And so DNA polymerase is like this, as you can see here in this picture. We have our DNA and DNA polymerase, and the clamp is also on the both side of the replication fork, right? But on the lagging strand, this will releases the sliding as soon as it reaches the 5 end of the preceding Okazaki fragment. The polymerase releases itself from the clamp. It dissociates from the, so as soon as it releases from the clamp, the DNA polymerase will dissociate from the template, right? And this polymerase molecule then associate with a new clamp that is assembled on the RNA primer of the next Okazaki fragment. Then it's again associated with the clamp to the next RNA primer of the next Okazaki fragment. So this is how it works. That is complicated, but this is how the replication process does. Process of DNA replication. So the replication machine has a DNA helicases, single-stranded DNA binding protein, DNA topoisomerase. Says we have a sliding clamp, we have a clamp loader, right? And this all move together. So this is the entire replication machines that move together to the entire DNA strand, right? So this DNA polymerase has our sliding clamp as well. So we'll just make like this, right? So this is all the entire process, right? So DNA replication first requires to open the DNA replication fork, right? We studied that. Then in order to start the replication, DNA needs RNA to prime. So that is a primase enzyme, right? That is needed to join the two nucleotides, right? For the leading strand, and for the lagging strand, it needs to make a short strand of the primer, and for that the RNA polymerase is used. And then once the primer is built, the DNA polymerase can take over and it continues to build or to replicate the DNA. Alright guys, so that was our replication machine, that is DNA replication process. So there is one more part for the DNA replication. So as we saw how the DNA is replicated. So but when the DNA replication reaches at the end of the replication, so then there is a problem with the lagging strand. It cannot complete the DNA strand. It cannot complete the DNA replication at the lagging strand, and how it completes we'll see in the next video. Alright, thank you guys for watching, and don't forget to subscribe.